Great. All right, so just as a reminder from last time, and this relates to what we were talking about earlier, what we have are, in Usilago and other fungi that mate in a similar fashion, you've got these cells of opposite mating type that reproduce by budding. So these are what we call haploid sporidia, and if they're compatible with each other, then they have differences at both the A and B mating type loci. So the A locus is involved in cell recognition. So these guys recognize that they're different based on the pheromones and pheromone receptors. You get conjugation between them. At this point, as you can see, there's now a mixture of the cytoplasms between these two. So whatever's being produced in here that we just talked about and is being produced in here, it can mix. That means if this one is producing, say, the B East and B West 1 versions, and this one is producing the B East and B West 2 versions, when they're together in this cell, if they're compatible like that, then you can get a B East 1, B West 2 heterodimer, and that will lead to the formation of this dikaryon. The dikaryon, we said, is filamentous. The two nuclei remain separate in the dikaryon, and that filamentous structure penetrates the host plant as shown here. And then that leads to, as we just saw, by the activation of the B locus, the subsequent steps that lead to formation of the diploid spores, so that we can recapitulate the cycle by the spore being released, germinating, undergoing meiosis. Okay, and then this just is a reminder of what we just talked about a few minutes ago. Again, the A locus, different in the A locus means that they produce a pheromone and receptor that are compatible, so you get cell fusion. And then subsequently, when the cell fusion happens, you've got the different B loci. So here's a B1 nucleus and a B2 nucleus with B West 1 and B East 1, and now B East 2 and B West 2. And in the same fused structure, you now got an active transcription factor, in this example, B East 1, B West 2 heterodimer, and that's a homeodomain type transcription factor that leads to sexual and pathogenic development. Okay, now, this is what we talked about before. I don't know how many of you have been interested in fungi, but one of the things that was rediscovered, I believe in the 1990s, was that yeast, which normally form a nice smooth colony on a petri dish, they will change the way in which they grow. So these are individual budding cells that have formed a colony. They will change the way they grow if you have a fermentable carbon source like glucose and they're starved for nitrogen. So normally you have bipolar budding. You can bud from either pole and you get cells that separate. But under these conditions of nitrogen deprivation, you get unipolar budding. So here's a mother cell, and she can only bud from this end, and those buds, they don't leave. So again, this is that clingy mother kind of, or clingy parent kind of a syndrome where they stay together in this chain and they produce what's referred to as pseudohyphal growth. Okay? And in Usilago, you can see different forms of this type of growth. So the mating type of growth you can visualize as this fuzzy appearance on a black charcoal background, charcoal agar background, whereas if you're not producing mating, then they look more faded like this. So these guys are not mating. This is an example of a successful mating. And that on the plate looks like this, but in the plant you get filamentation that looks like this. So there's actually branched filamentation going on in the plants that are infected. However, this is another interesting phenomenon, and that is that Usilago, this is a haploid colony. So these are haploid cells. Normally we said that they would produce a nice smooth colony on rich media, but if you starve them for ammonium or other nutrients like nitrogen that are not necessarily ammonium, they produce these filamentous structures, and the cells themselves look filamentous. Okay. Now, next slide. Okay, actually go back. Okay, this particular ability though, to switch from a yeast-like form to a filamentous form, or from a filamentous form to a yeast-like form, is important in terms of disease causation. So those of you that are more biomedically oriented and don't really care about plant pathogenic fungi might be saying, so what? And the so what is that that transition is required for a number of different kinds of fungi to be pathogenic. So I don't know if any of you've heard of Candida albicans. You've heard of it, you know what it is?
Isn't it like thrush?
Yeah, so Candida albicans is a causal agent of thrush. So it's a yeast, somewhat distantly related to Saccharomyces. And it's also involved in yeast infections that females are usually, not usually, but can be affected by. How about Cryptococcus neoformans? Maybe not. It turns out that Cryptococcus neoformans, if we were to go around the room, or when it was safe to do so, if we were to collect tissue samples from the lungs of people in the Ohio River Valley, we'd probably find a fair amount of spores, or not spores, but sort of places where the fungus has infected the lung, but it's not active. And that particular fungus, when it becomes active, it is able to cause fungal meningitis. So Cryptococcus neoformans is responsible, it's the largest cause of fungal meningitis in the world. Okay? But it can only do that in most cases, most of the strains that we're exposed to, you can only do that if your immune system is disrupted. Because if your immune system is disrupted, what will happen is Cryptococcus, which normally grows as a yeast-like phase, will switch to a filamentous phase to cause disease. So that transition between, and we already know that for Eusilago it's important, and some other fungi as well. So it's important for us to understand how that transition takes place, particularly if you're interested in diseases that are caused by fungi. Okay? All right, so we're going to talk about Eusilago and Saccharomyces, and also sort of briefly bring up another smut fungus, Microbacterium violaceum. Okay? Microbacterium violaceum, if you recall, is another of those smut fungi that we talked about earlier. It's a fungus that infects flowering species in the carnation family. And it has a life cycle that's more similar to Eusilago matus. It's not identical because it doesn't produce tumors, but it has a number of aspects in common. Okay? All right, so we just finished talking about the cyclic AMP dependent protein kinase A pathway. And both Saccharomyces and Eusilago have this pathway. Both of them have a regulatory subunit that blocks the catalytic subunit, and cyclic AMP is responsible for pulling off the regulatory subunit so the catalytic subunit becomes active. In Saccharomyces cerevisiae, this pathway becomes active under conditions of low ammonium. Now we're talking about ammonium because ammonium is probably the most accessible form of nitrogen for a lot of organisms. And so if you have ammonium available, that's a nice way for the cells to get their nitrogen content. And what we see here is that Saccharomyces cerevisiae, when conditions of ammonium are low, it produces filamentous growth. Now why would the fungus do that, you might ask?
Why would the fungus do that?
Yeah, why would you did?
Oh.
Yeah, why would they?
Sorry. Just growth in low ammonium conditions. Is that like in the starvation condition?
It is a type of starvation condition, but why would they switch from yeast-like to filamentous?
Are they trying to go out and like attack and find other nitrogen sources?
Yeah, it's kind of their way of scavenging for nitrogen that might not be where they're located.
So
See,
they're like nitrogen.
Yeah. Is there any nitrogen out there? I mean, if you think about it, yeast cells on an agar plate, they're not motile. Right? When they land on the plate, they're stuck there. They can grow and divide, but normally you get a nice discrete colony on the plate. So if that's the case and there's no ammonium, they're stuck, right? Okay, we're gonna starve, we're dead. But if they now switch to this filamentous growth, that means that they can extend out their cells and look for a new place where there might be nitrogen. I mean, at least that's our human interpretation of why they might want to do that. Okay? All right. Now, Eustalogomatus, we sort of indicated that on nutrient-rich conditions, they normally are budding. Okay? And what's interesting about this, particularly from the standpoint of people that are interested in evolution, is this happens when, again, the pKa is active. So remember, for Saccharomyces, when pKa is active, they're filamentous. When pKa is active in Eustalago, they're budding. Okay? So they're using this same pathway. And this pathway, by the way, I didn't really emphasize this, but these pathways we've been talking about, they are conserved all the way from fungi or other microbes all the way up to humans. So there's MAP kinase pathways in fungi, there's MAP kinase pathways all the way up through to humans. Similarly, there is the cyclic AMP-dependent protein kinase A pathway in fungi, but again, all the way up through organisms, including humans. So what's happened is these two fungi, they both make use of the same pathway, right? They're making use of cyclic AMP, adenylate cyclase, the regulatory subunit, and then the catalytic subunit. But what's happened is the output is different, right? So the output is different, and in this case, the input, it's the same thing that's being assessed, availability, but opposite in terms of which signal leads to which form. So in Saccharomyces, low ammonium is the outside signal that leads to filamentous growth. In Eustalogomatus, again, assessing nutrient conditions, nutrient-rich conditions, again, lead to budding growth, but in both cases, the pKa is active. Okay? Right. Oh, yeah. Oh, thank you. Sorry. Yeah. So this is the addition. The addition is, what is it that's sensing availability to ammonium? So remember here for Saccharomyces, the availability of ammonium is what's controlling how this works. And it turns out that there is a family of proteins that are transmembrane crossing proteins. In fact, and this one I think there are 11. 1, 2, 3, 4, 5, 7, 8, 9, 10, 11 transmembrane domains for these kinds of proteins. And what these proteins are involved in is the transport of ammonium across the membrane, okay? And so what we're going to talk about is what are these proteins and what role do they play in this particular signaling pathway. This group of proteins are referred to as either ammonium transport proteins or methylammonium permease proteins, okay? In yeast, they're referred to as MEPs, or methylammonium permease proteins. But this is a family of proteins that's conserved from bacteria to humans. So humans have an ortholog or homolog of this type of protein that's found in yeast and bacteria. And it turns out that that ortholog is the Rh factor that's found in our red blood cells. So it's in that same family. And if you take that protein and you put it into yeast that are defective for their protein, the human protein will correct for or complement that defect. So functionally, they are conserved in many respects as well. Okay? All right, so in Saccharomyces, and that's what these particular genes refer to, you have 3 different ammonium transporters, MEP1, MEP2, and MEP3, okay? All of them are involved in the transport of ammonium. And if you delete all 3 of them, what happens is those mutants of yeast are unable to grow on low ammonium. And by low ammonium, let's just say about 50 micromolar. I mean, in reality, less than about 1 millimolar, and they won't be able to grow. So they're severely defective in growth under low ammonium. And what you can do is, in the same way that we talked about complementation when we were talking about assessing the components of Eustalago for the UBCs, you can take individual genes and put them into these mutants to see can they fix the problem. And so if you take any one of these, so you take this triple mutant and you put into it a functional copy, say, of MEP1, it will allow those cells to grow on low ammonium. If you take just a copy of MEP2, it will allow those cells to grow on low ammonium. If you take a functional copy of MEP3, it will allow the triple mutant to grow on low ammonium. However, only MEP2, when you put that into the triple mutant, not only does it allow them to grow, but what else does it allow them to do?
Fermentation.
It allows them to do filamentation. It's the only one that allows that filamentation phenotype to come back. So remember, Saccharomyces on low ammonium is supposed to filament. The triple mutant doesn't grow on low ammonium, and if you put in MEP1 or MEP3, even though it grows, it doesn't filament. So MEP2 is the only one that allows for that filamentation to take place with the triple mutant. And it seems then that MEP2 is the one that's required for the ability to sense how much ammonium is there. Because we know that the response to sensing nitrogen or ammonium availability is in Saccharomyces to filament. Okay? So MEP2, we think, is a different kind of transporter. It's not just a transporter, it's kind of also a receptor for sensing how much availability of nitrogen in the form of ammonium there is. And so the new term for a protein like MEP2 is something called a transceptor. So it's a transporter and a receptor in one. Okay? Now, that's all very interesting, but we're also interested in the smut fungi. And so the microbotrium, so the anther smut, we're interested to see what about its ammonium transporters. And so we isolated the ammonium transporters, one of them anyway, the ammonium transporters from microbotrium, and we put the microbotrium gene called MEP-A into this triple mutant of yeast to see if that protein could fix the problem of the yeast. So first question is, does that protein allow the triple mutant to grow on low ammonium? Yes or no?
Yes.
Yes, that's right. We have a unanimous vote on that one. Yes. So MEP-A can fix the ability to grow on low ammonium problem, but does it fix the filamentation problem?
No.
No. Unanimous again. So MEP-A is a transporter, but it isn't able to help in the sensing of ammonium availability. Now we look at Euselago. Euselago actually has two ammonium transporters that are named UMP1 and UMP2. So UMP1, when we put that into the triple yeast mutant, does that allow the triple yeast mutant to grow on low ammonium?
Yes.
Yes. Yes. That was a question or an answer?
Yes.
Yes. Yes? Okay, yes, it does allow it to grow. Does it allow it to filament?
Yeah.
Nah, not really. You wouldn't bet your money on this one. Yeah, it looks like it's not really. So that would suggest that UMP1 again is an ammonium transporter, which we have determined that it is, but it's really not able to help in the sensing aspect. Okay. What about UMP2? Does UMP2 correct the growth defect on low ammonium?
Yes.
Yes. Does it correct the filamentation problem?
Yes.
Yes. So UMP2, we think, is a functional homolog or ortholog of MEP2. So now we have a Euselago protein that does, at least in yeast, the same thing that MEP2 does. So that's kind of cool. Okay? Any questions? No? Okay. All right, well, I have a question, and that is, if that's true in yeast, is it also true in Euselago? In other words, does UMP2, is UMP2 required for filamentation in Euselago? Well, we may have mentioned this earlier, but the fact is that wild type Euselago on low ammonium, these are haploid cells now. They are not producing a nice smooth colony. They are producing a fuzzy colony because on low ammonium, they are able to then produce these filamentous structures with the idea that maybe they're out looking for a new ammonium source. Okay, so that's what wild-type does. If you knock out or disrupt the UMP2 gene, what's happened to growth on low ammonium?
No fermentation.
Yeah, no filamentation. They're still able to grow, so it's not required for their growth on low ammonium, but it is required for this filamentous response with respect to low ammonium. Okay, so that's consistent with at least our current working hypothesis for how it works. Yes?
So the only thing they would have would be the UMP1 transporter.
That's correct. That's right. And so the question that was asked is, do they have the UMP1 transporter that's working? And the answer is yes. And then maybe to follow up on that question, whether you were going to ask it or not, it turns out if you knock out UMP1 just by itself, there's no phenotype. We have not been able to identify any phenotype of just knocking out UMP1 by itself. So UMP2 is critical in terms of the ability to produce these filamentous structures on low ammonium. Okay? Next. Okay, so now the question is, what is it that makes some ammonium transporters able to be involved in this sensing process and others not? And this particular example, I believe, so SPG, yeah, so this particular example is what's known about the structure of the ammonium transporter from E. coli. So AMTB is an E. coli ammonium transporter protein, and it's got this GKPS, which is shown here, GKPS. This is the inside of the cell, this is the outside of the cell. And as you can see, this is a protein that spans the membrane, and this is where this region would be in bacteria. Now bacteria, E. coli, they do not filament in response to low ammonium. This is something that fungi do. In fact, as far as we know, those are the only cells that respond by filamentation. And then the question is, okay, so how are these ammonium transporters from fungi different from this ammonium transporter from E. coli? Well, looking at this region, what you have is these four residues, arginine, lysine, tryptophan, and tyrosine, I believe. No, excuse me, threonine, or lysine, tryptophan, and serine. Those residues form what's called a target site for, guess which enzyme? PKA. So PKA recognizes this site, not the one in E. coli, but it recognizes this site in proteins, and it typically will phosphorylate the threonine or serine that's present in that location. Okay? And so now we're trying to see, is there a connection between the PKA pathway, which we saw earlier, there seemed to be something involved in that pathway, and the function of these ammonium transporters. Okay? If you could click again. So this illustrates where this would be. We're using the bacterial one, but if you think about just the K, W, and S, so this part here, RKWS, wherever that is, here's one. This would occur right here, just at this side of the membrane, inside the cell. And so the question is, if that were phosphorylated, say by PKA or not, would that have an effect on the way the protein works in terms of sensing ability? So, next slide. Okay, well, it turns out that if you change that PKA site, that putative PKA site, to a site that no longer can be phosphorylated, and the way that you do that is you change the serine or threonine to an alanine. That amino acid cannot be phosphorylated, and then the question is, how does that affect the function of the protein? Well, it turns out that making that single amino acid change allows for ammonium transport, but abolishes pseudohyphal growth. And we'll see that on the next slide. So, this is the yeast triple mutant again. If you put in MEP2, remember, not only can they grow on low ammonium, but they filament. if you make a mutation in the yeast ammonium transporter, MEP2, from a threonine to an alanine, that cannot be phosphorylated by pKa, and what happens to those cells?
No fermentation.
No fermentation, but they still can grow on low ammonium. So transport has not been affected, but the signaling has been, okay? Suggesting that the ability to be phosphorylated in this position is critical for function. Now, some of you might be skeptical, which you should be as scientists. And you might say, well, what might you say as an alternative to saying that T specifically is important?
Just the A is important?
Well, making that change to an A eliminates that function, but what could you argue? I say it's because it has to be phosphorylated by pKa, and you say
It could just lead to a misfolded protein.
yeah, it could just be a misfolded protein. It could be if we put in anything here, right? If we put in any other amino acid, that could screw up the function. That's a very good thing to ask. Well, serine is another amino acid that can be phosphorylated. If we make the change from threonine to serine in that position, what happens?
Fermentation is restored.
Yeah, or to put it a different way, making that change doesn't change the phenotype. So not only can they grow on low ammonium, but they also filament, which suggests that what's important is that in this particular position, 288 in this protein, you have to have a residue that can be phosphorylated. My guess is if we put in any other amino acid that can't be phosphorylated, it would also disrupt and give us something that looks like that. Question?
Just out of curiosity, is there, like, does it take longer for the fermentation to develop, or?
No. No, these are all same age colonies. Good question. All right, so now the question is, how does this apply to Eusulago? So now we do the same thing with Eusulago with UMP2. Again, the triple mutant of Cerevisiae, if you put in UMP2, you get not only growth on low ammonium, but also fermentation. However, if you take that region in UMP2, which has a serine in the critical position, and you change that serine to an alanine, they still grow, but notice there's no fermentation. Okay? Next slide. Okay, so our conclusions are we've got 3 of these SMUT ammonium transporters, 1 from Microbacterium and 2 from Eusulago. All 3 of them are able to complement the triple mutant of yeast that has all 3 of its ammonium transporters disrupted. They can all grow, any of those can allow growth on low ammonium, but only UMP2 can complement the pseudohyphal growth defect in yeast. Furthermore, in Eusulago, UMP2 is required for filamentous growth on low ammonium. All right, now, there's several lines of evidence that suggest a connection between the ammonium transporting ability and the pKa pathway. And first of all, Saccharomyces cerevisiae MEP2 mutants do not filament on low ammonium. But if you add cyclic AMP, so in other words, if you stimulate the activity of pKa, then they become filamentous, okay? And that's shown here. So here's wild type. Here's the mutant that doesn't have MEP2, but if you add cyclic AMP to that mutant, you get filamentation, which tells you that activating the pKa pathway in Saccharomyces is a way to get around the fact that you don't have this ammonium transporter. Okay? Now, you get a similar set of conclusions from Eusulago, but you have to keep in mind and remember that in Eusulago, when pKa is active, you get budding, and when it's inactive, you get filamentation. So in this situation, normally on low ammonium, you get filamentation, but if you add cyclic AMP in order to block pKa, in order to activate pKa, you get budding growth instead. So the colony is smooth and the cells are budding. And that shows you again that the activity of pKa makes a difference in terms of the response to low ammonium. Okay? All right, so kind of recapping, you've got this pathway, the cyclic AMP dependent protein kinase A pathway, where the production of cyclic AMP affects the regulatory subunit of pKa, allowing for pKa to become active. And now we're showing that this ammonium transporter is somehow involved. And the key is that what it looks like is, in order for this to work properly, in order for the ammonium transporter to carry out its function appropriately, you need to have an active pKa, and you need to have the ability of your ammonium transporter to be phosphorylated by pKa. Is that it? That's it. Okay. And that is it for today's presentation.